Hey everyone, welcome back. In this video, I want to go over the stages of cellular respiration. There's 3 of them, and we're going to talk just kind of briefly about what happens in each. They are pretty complex when you look at them in depth, and in this case, I just want to kind of give you guys an overview of what's going on in each so you can really get a good idea of how cellular respiration works, how the energy from sugar gets released, and how that energy is then used to make adenosine triphosphate. And in some previous videos I told you a little bit about just the chemical reaction of cellular respiration, how we're using glucose and oxygen and through a series of reactions breaking that down, and a byproduct that gets produced is carbon dioxide and water, and then the energy that's being used or being produced here is adenosine triphosphate. So it's why we call the mitochondria the powerhouse of the cell. It's really taking the carbohydrates that we eat, we use those for energy, and we use that energy to make ATP, which is what cells use for energy. So in some other videos I go into more detail about how ATP gets used and the chemical reactions for cellular respiration, and if you're still confused a little bit about that, please go back and watch those. In this one though, I want to talk about the stages of cellular respiration. So let's go ahead and take a look really quick at what these are. There's 3 of them. And so in this image here, it's just kind of a brief overview, kind of 30,000 foot view of what's going on here. The first stage here is called glycolysis. Glycolysis actually happens outside the mitochondria, and in that process glucose gets broken down initially into these 2 molecules called pyruvates, and in that series of reactions, or those reactions that happen there, ATP gets used. So there's 2 ATP molecules used, energy goes into that to help break this apart, and it actually produces 4 total ATP, and since 2 of them are used and 4 of them get produced, we have a net gain of the 2 ATP molecules. As these chemical bonds get broken, there's energy released in the form of electrons, and these high energy electrons are going to be used later on down the line, and they get collected by these electron carriers. In this case it's NADH. NADH is a molecule that harnesses those electrons and brings them down over here to the electron transport chain. We'll talk about that in a little more detail later. These pyruvates then enter the mitochondria, and once they do that, they go into this inner matrix inside the inner membrane here, and they go through a series of pretty complex reactions referred to as the Krebs cycle. That Krebs cycle then breaks those down even further and produces again 2 more ATP molecules, so we have a net gain of 4 by this point. And as those chemical bonds get broken, again more electrons get released. Those electrons get carried over here by NADH again, and a new one called FADH2. And again, these are just molecules that are bringing electrons that are being released from these reactions over here to the electron transport chain. The electron transport chain and oxidative phosphorylation are a series of steps that take place across the inner membrane of the mitochondria. We'll go into some more detail in a little bit on that, but the end result of this is the production of anywhere between 32 and 34 molecules of ATP. And really this number depends on the molecule that's bringing the electrons over to the chain. Each one of these is going to bring electrons to a different place in that electron transport chain, and because of that it can either produce more or fewer ATP molecules. All said and done though, we're looking at anywhere between 36 and 38 molecules of ATP for every glucose molecule that gets broken down inside, right outside and then inside the mitochondria. So let's dive in a little bit deeper for each one of these. Stage 1, glycolysis. Up here at the top of the slide I have the general equation for cellular respiration so that we can kind of refer back to it. Just to go over this, we have sugar, in this case glucose, we have 6 molecules of oxygen, and those 2 are going to enter through these reactions and then ultimately produce molecules of ATP and give off some byproducts of carbon dioxide and water. And really when we talk about respiration, we're breathing in oxygen and we're breathing out carbon dioxide, and this is kind of how that works. So, glycolysis, what is it? When we break this word apart, glyco. Glyco means sugar. And so we're talking about sugar here. And then this word lyse, that root word lyse, means to split or to break apart. So looking at the word that way just really means the splitting of sugar, or in this case, the splitting of glucose. So glycolysis translates out to splitting of sugars. Glucose is a 6-carbon molecule. If we split that, a really basic way of looking at that is we're going to produce some 3-carbon molecules. It's a really basic way of thinking about it. It's a little more complicated than that, but for this video I just want you to understand we're taking glucose, splitting it apart, and we're creating these 3-carbon pyruvate molecules, sometimes called pyruvic acid, and those molecules then are going to enter the mitochondria. It takes energy to do this. So the cell is going to use 2 molecules of ATP to make that happen. Ultimately, glycolysis is making 4 molecules of ATP, but since we're using 2 of them to make this go, it's a net gain of 2 ATP molecules total. If we look up here, we see that these electrons are going to be carried by this molecule called NADH. I don't want to go into a whole lot of detail about what that is and how all that happens. What I want you to know for this video is that as we break chemical bonds, energy gets released really in the form of high energy electrons that get shed from this whole thing. And those electrons then are going to be picked up by these molecules, these electron carriers, in this case NADH, and those are going to be carried over to the electron transport chain where they get used later on, and we'll talk about that as we get to it. So going back to our reaction here, we're using glucose here. So this is our first reactant that gets used. Glucose gets split, cut in half basically, to form the pyruvates, and we're producing a few of these ATP molecules that we're going to end up with as long as everything runs as smoothly and as completely as it can. So that's the start of cellular respiration, glycolysis, and it happens outside the mitochondria. Once those pyruvates are formed, once those the glucose gets broken down, they enter the mitochondria. They go into the inner matrix of the mitochondria. So they pass through the outer membrane and through the inner membrane, and they go into the inner matrix of the mitochondria, and they go through a series of reactions. It's a really complex cycle of reactions, too complex to go over here. But what I want you to know is the Krebs cycle is those series of reactions. In that, more ATP is produced. We're going to produce 2 more molecules of ATP. Carbon dioxide also gets produced in this series of reactions. As we're breaking those pyruvates down even further, some of the carbon that gets released from here, some of the oxygen that gets released from here is going to produce CO2, and that's what we breathe out, and that's again how we get the name cellular respiration. As that's happening, as all these bonds are being broken and all the energy is being released, it's being released in the form of electrons, and we have NADH show back up and collect some of those electrons to bring over here. And we have a new one here, FADH2. And so these 2 electron carriers are going to harness some of those electrons, some of those high energy electrons, and bring them over here to be used in the 3rd step. Again, really the Krebs cycle is just breaking those pyruvates down even further, breaking the glucose down even further, and releasing more and more of that energy. So going back to our reaction here, we're still working with glucose, we're breaking it down more and more, we're producing 2 more ATP, so we've got a total of 4 now, and this is where the CO2 comes from this chemical reaction. Let's take a little bit closer look into the Krebs cycle. It's a really complicated picture here, guys. I don't want to go into all of the detail for this one. What I want you to see is that it is this really complex series of reactions that are taking place. But ultimately what we have are the pyruvates coming in, and these pyruvates just continue to be broken down further and further as they move along. So we're breaking those bonds between the carbons and releasing the energy there. And so some of the energy that gets released gets picked up in the form of electrons by the NADH. Some hydrogen ions are released, and those are going to be important as we look into the next stage. We have carbon dioxide being released, and so CO2 is coming out of this. Here we have some more CO2, some more NADH grabbing those electrons and carrying those to the next stage. continues all the way down. We're producing more CO2, more electrons. All of this energy gets released as we continue to break those pyruvates down. So we're breaking it down farther and farther. So that's a really simplistic overview of the citric acid cycle, or the Krebs cycle, but ultimately what I want you to know from this is we're just breaking the sugars down even further. So stage 3, the electron transport chain. This is where we're going to produce the majority of our ATP, and it really depends on which of those electron carriers are bringing the electrons in, whether it's NADH or FADH2. This is also where the oxygen gets used. And so all that energy that we've harnessed in those electrons that are being brought over here to the electron transport chain then is going to be used to produce, or really to power, some transport across this inner membrane, which ultimately supplies the energy for an ATP synthase enzyme. Let's take a little bit closer look at that, and I can explain it in a little more detail here. So here we have our inner mitochondrial membrane. Down here is the mitochondrial matrix. So this is deep inside the inner parts of the mitochondria. This area out here is the space between the inner and the outer membrane. We have a series of proteins that are along this membrane here, and these proteins are really, the job of these are to move hydrogen ions across this membrane, and so it's feeding hydrogen ions into this space between the inner and outer membranes of the mitochondria. Ultimately, this is going to create a concentration gradient where we have a lot of hydrogen up here and very little hydrogen down here. So if we're moving hydrogen ions from low concentration to high concentration, it's going to require energy to do that. And this is active transport, going back to some cellular transport things that we've learned in the past. To do that, the energy that's been brought over by NADH in the form of electrons is being supplied to these proteins. And so when that energy gets dropped off here, when those electrons get dropped off, that provides the energy to pump these hydrogen ions into this space between the inner and outer membranes. And so we're filling this area with hydrogen ions. NADH is going to drop their electrons off here at this first protein, and once that protein uses those electrons, they actually get transferred over here to these series of proteins here, which then move it over to this third protein, which is also going to be a method of transporting hydrogen across. And so these proteins here are just really moving the electrons from one protein to the next. FADH2 will drop its electrons off here. Since it's dropping them off here, it's only moving a few hydrogen ions across. It doesn't get to, the electrons don't get to pump hydrogen ions here, they're only pumping them here and then ultimately through this one. So FADH2 is moving hydrogen ions across two proteins. The energy that's brought by NADH is actually supplying energy for three proteins. So if we get more NADH there, then it actually builds up a bigger concentration gradient of hydrogen here, which ultimately can produce more ATP. That's kind of how that happens. But what's going on is the electrons are moving down these proteins and pumping hydrogen into, providing energy, to pump the hydrogen into this space. As that concentration builds up, these hydrogen ions are going to want to diffuse back across the membrane, and that's where this protein comes into play. And this is a big complex of proteins and an enzyme called ATP synthase. And it allows the hydrogen ions to diffuse back across. It's facilitated diffusion, and they want to move this way because, again, there's a high concentration of hydrogen up here. So they diffuse across this and provide energy for this enzyme ATP synthase to add that third phosphate group onto ADP. And that's a high energy bond, so the energy that the hydrogen produces moving across this feeds that ATP synthase enzyme, which allows us to build the ATP molecule here, which the cells ultimately use for energy. And so all said and done, we're going to produce anywhere between 32 and 34 ATP molecules from this. Overall, here's just a quick overview. You guys can read through this. I don't need to read it for you, but you can look at that and see exactly kind of what's used and what's produced in each one of these stages. So it kind of goes over where each one of those reactants and each one of those products are used and produced. I hope this helps clear up some of those stages. I'll go into some more detail later on.